today along with my degree students, as B.Pharma students, I am here to give you the demonstration with respect to the qualitative test for carbohydrates. As all of us are giving that carbohydrates are the polyhydroxyaldehydes or ketones or compounds obtained after hydrolysis of these. So there should be some methods which can be used for identification of whichever the sample is given to you as a carbohydrate. For that reason, there are some assigned tests are there, qualitative tests are there, which we can carry out to identify which is the sample given to us. That is, whether it is a monosaccharide or disaccharide. After getting to know that, we can even get to know that what exactly that particular compound is, whether it is a glucose or fructose or starch or whether it is maltose or lactose. So there are some series of tests are there which we need to carry out. So coming to the different tests, so we are having the different tests like Mollis test we are having the solubility test, we have got iodine test, then Benedict's test, then Felling's test, then Barfoed's test, then Selivanov's test, then the Osazone test. So by performing these tests in a systematic manner, we can get to know that which is the sample given to us, whether it is a glucose or fructose or maltose or lactose or starch. So coming with the different tests importance, when we perform a Mollis test, we can get to know that whether the given sample is carbohydrate or not. Okay, if the given sample is a carbohydrate, then our next procedure is to identify which is that. So during that process, we go for the solubility test. If in the solubility test, if we get to know that if the compound is soluble in the distilled water, then it shows that the compound is monosaccharide or a disaccharide. If it is insoluble, it shows that the compound is a polysaccharide. Then the next test is iodine test, which will help us to find out whether the given sample contains starch or not. Okay, that is a polysaccharide results. Then there are two tests like Benedict's test and Felling's test, which will enable us to identify whether the given sample is reduced in sugar or not. Then there is a test, that is Barfoed test, which will enable us to get to know that whether the given carbohydrate sample, whatever is given to us, whether it is a monosaccharide or a disaccharide. Then coming to the next test, that is Selivanov's test, it will give us the information that whatever the sample is given, whether it is a aldose containing, that is whether it is an aldose or a ketone, that is aldehyde as a functional group containing carbohydrate or ketone group as a functional group containing carbohydrate. Then comes the finally the Osazone test. It is basically is also called as vinyl hydrazine test, which will enable us to get to know that whether the given sample is a glucose or a fructose or a maltose or a lactose. That is, it is a final confirmatory test. How we are doing that? Our students will explain the same thing, and the crystals, based on the types of the crystals formed, we will be able to tell whether it is a glucose or a fructose or a maltose or lactose. For example, for glucose and fructose, we get a yellowish needle-shaped crystals, whereas for maltose we get the powder puff-shaped crystals, whereas for the lactose we get a yellowish ball with the needle with the such type of the crystals. Okay, with this brief information, now I will call upon my students to give the demonstration with respect to the identification or the qualitative test for the carbohydrate.
Greetings to everyone. Myself, Rakshita Sugar. I am performing the qualitative test for carbohydrates for the unknown given sample. First one, Mollis test. 2 ml of test solution and 2 drops of Mollis reagent plus 1 ml of concentrated H2SO4 sides of the test tube. By observing it, we will get the violet ring at the junction of two liquids. It indicates the presence of carbohydrate.
Performing the first test, Mollis test, adding 2 ml of the test solution, then adding 2 drops of Mollis reagent. Now we need to add 1 ml of concentrated sulfuric acid along the side of the test tube.
Solubility test. Few mg of sample plus 2 ml of water. While observing it, soluble in water. Inference indicates the presence of monosaccharide and disaccharide. Solubility test.
In the second test, solubility test, we will be adding 2 ml of water to 2 ml of the sample solution. As we can see, it's soluble in water.
Third step, iodine test. 2 ml of test solution plus 2 drops of iodine solution, boil for 2 minutes. By observation, no appearance of blue color. Starch is absent.
Is the iodine test. To the 2 ml of the test solution, we will be adding 2 drops of iodine solution.
Benedict test. 5 ml of Benedict reagent plus 8 drops of test solution, mix well and boil and keep it for cooling. By observing it, appearance of orange PPT, inference, presence of reducing sugar.
The fourth test is the Benedict's test. To the 5 ml of Benedict's reagent, we will be adding 8 drops of the test solution. Now we will mix it well and boil for 2 minutes.
Fifth step, Fellings test. Take 2 ml each of Fellings A and B solution, 1 ml of test solution, boil for 2 minutes and keep it for cooling. By observing it, reddish brown PPT indicates the presence of reduced sugar.
Now the fifth test is the Fellings test. We will be taking 2 ml each of Fellings A and Fellings B solution. Plus 1 ml of the test solution and we will boil it for 2 minutes and let it cool.
Step, Barfoed's test. Take 1 ml of Barfoed's solution plus 1 ml of test solution and boil for 2 minutes. By observing it, we will get the red PPT, inference, presence of monosaccharide.
The sixth test is the Barfoed's test. We will be taking 1 ml of Barfoed's reagent plus 1 ml of the test solution and boil it for 2 minutes.
Seventh step, Selvinov's test. Take 1 ml of Selvinov's reagent plus 1 ml of test solution, boil for 30 seconds. By observing it, we will get red PPT, inference, presence of aldosis.
Seventh test is the Selvinov's test. To 1 ml of test solution, we will be adding 1 ml of Selvinov's reagent. And we will boil it for 30 seconds.
Osazole test. 5 ml of sugar solution plus 0.3 gram of osazole mixture plus 5 drops of glacial acetic acid boiled in a water bath for 30 to 45 seconds. Allow it for cooling and observe the crystals under the microscope. By observing it, we will get the yellow needle shaped crystals. Presence of glucose. The report. By performing the qualitative test for carbohydrates, it was found that the given sample indicates the presence of glucose, which is monosaccharide.